Hello! Today I'm here to talk to you about the research studies that investigate how amphetamine exposure in C. elegans causes the reduced uptake of dopamine and future developmental deficiencies. This is pertaining to the research studies that were completed by McCohen et al. And Carvelli et al. In these studies, what was done is they used C. elegans that are DAT1 line, GFP, green fluorescent protein lines, which means they have a protein that will illuminate inside of them when it is exposed to a fluorescent light source, like inside a microscope. There is a light source mode called GFP, and this is used for visualizing these kind of organisms, these kind of animals that have this green fluorescent protein. In these studies, what they were looking to study is how amphetamine affects an animal itself, using the C. elegans, for example. What they did was they took amphetamine at a solution of 0.5 millimolars, and when they were incubating the C. elegans inside tubes, typically they're incubated for 15 hours, they would add amphetamine to the experimental group and then have a control group which is just M9, and it would be left for 15 hours, visualized at 10 hours, but fully grown, and they hatch out of the embryo, the offspring come out and hatch at 15 hours. But in this study, we visualize what is going on in the developmental process. And we also completed behavioral assays. In these behavioral assays, the C. elegans were taken at a certain stage and put into different wells. One well had sucrose, which is our control solution, and one well had amphetamine. In our sucrose control solution, there should be no SWIP. SWIP is swimming-induced paralysis. What that means is when the worms are paralyzed, they stop moving. And in the behavioral assay, we take the amphetamine pre-treated line and put it inside the well with amphetamine as well as sucrose. And then the control group, which is just normal C. elegans, those are put into the sucrose as well as what they call acute amphetamine exposure tests. And that means these worms are put in the amphetamine solution as well, even though they were not pre-exposed. Then for 10 minutes, every 1 minute, we record to see if any of them SWIP, swimming-induced paralysis, if they stop moving. And I'm going to show you right now, and continue to talk and explain, some imagery of what is going on inside the C. elegans itself, what you can see, and what those pictures and visualization and different markers you will see in the pictures tell us as researchers, and how it explains what is going on inside the C. elegans itself. Hello! So right now you're able to see a C. elegans. Amphetamine increases the excitatory neurotransmission and monoamines in the brain. The reason that amphetamine was investigated is specifically to find out what developmental effects this drug can have on people and what mechanisms cause these effects. Now I know we're not using people today, but these C. elegans are good models to predict what could be going on inside humans as well. Now, such research is very important to study these psychostimulants, because these psychostimulants are, they're very addictive and they're often abused drugs. And by investigating the mechanisms of such a drug and how it affects the certain functions of, the animal, in this case the C. elegans, especially in this study, the results that were obtained from the research are very interesting and can be used to find ways to prevent the harmful effects and prevent future generations from being affected. Now, here you can see, this is a time lapse actually, this is in the early stage of the time lapse, and what the microscope does is it takes pictures of different layers of the C. elegans. So I will show you the different layers, and you'll be able to see. You see how the green fluorescence comes. This is about uh, halfway inside the body of the C. elegans, and then now the microscope is going higher up, higher up, higher up until the fluorescence is gone. So these are the places where the fluorescence is seen the most. And the reason why in the study they studied the C. elegans from embryogenesis, when the egg is birthed, I guess you could say, from the adult, all the way through the adult is and future generations, is not only was this study looking at the effects that can happen in the moment, in the of the first exposed uh, animal that is first time being exposed to such the drug, but they're also looking at the transgenerational effects. And throughout the experiment, they propagated the same lines. So they use the same lines and just keep reproducing them to continue to use them. And it was found that specifically through the generations, for the line of amphetamine exposed ones and then the line of the control group, that the development and the behavioral assays were all different and uh, unusual compared to what the control group is. Even in lines, in the amphetamine lines, second, third, or fourth generation, it was still showing the results according to the study as if they had just been exposed for the first time. So what that tells us is that the genetic inheritance can transfer and predispose these effects to future generations. And not only does it affect the genetics of these animals, but what exactly is going on is, according to application from what I learned in bioprinciples, there's a chemical modification of the histones that is directly affecting the regulation of gene transcription. And when I say this, I mean that there is a specific change in histone methylation. Now, according to the study, the amphetamine exposure during the development incubation period, embryogenesis, has shown to affect specific enzymes that cause a change in the histone methylation. This, in turn, causes the modification of the dopamine transporter genes, for example, DAT1. And this causes genetic modification to be passed down to future generations. This explains why traits that were seen in the originally exposed line of worms is continued to be seen and expressed in the future generations that are propagated, also known as the ancestral effects. And this is the effects that are seen in their predecessor line, like, father, grandfather, great grandfather, but we're talking about worms here. So the effects that were seen by, for example, the amphetamine line were also seen in the future generations, the original amphetamine line. Now, next I'm going to show you the embryogenesis visualization and finish up with the conclusion of the research study. Here you have the multiphoton visualization of, on the bottom two on the left, which look like hollow ovals. Those are the amphetamine exposed or pre-treated embryos. On your top right, which you'll see right here, some green GFP indicators, those are our M9 control group embryos. This is the first time this has been done, as this has not been visualized by any other laboratory. So here I will show you how it starts off, as this is a time lapse. So it starts with the growth. You see that there's already starting to show GFP indicator, while our experimental group is yet to provide any GFP indication. And they continue to grow. Yet, the amphetamine exposed embryos have yet to develop their dopaminergic neurons. So this is a very important piece of the puzzle. Not only does this show us that amphetamine causes developmental changes to the embryogenesis slash incubation period and process itself, but it also shows us that there are physical changes that the amphetamine causes, not only in their behavior once they have hatched and are adults or fully grown offspring, but amphetamine is also affecting them at the cellular level as their central nervous system is developing during their incubation slash developmental period. The conclusion of these studies has come to that amphetamine exposure during the embryogenesis process has shown to affect specific enzymes that cause a change in the histone methylation. This results in the modification of the dopamine transporter genes, like we talked about before, DAT1. Thus, this is causing the genetic modification to be passed down to future generations. And this is why the behavioral assays still show that future generations express the traits of the originally exposed offspring, such as these embryos, yet they have only been propagated and continue to um, hatch and continue to breed, I guess you could say, but they have not been manipulated in any way. They just come, they're just offspring that come originally from the same line of these embryos you can see on the screen. And this offers an explanation to why the behavioral effects observed in the original experimental group of worms were also able to be observed in the future generations. Now, in my opinion, from what I learned in bioprinciples, this is what answered the question to what causes future generations of progeny to express the ancestral effects they were originally expressed in their past generational relatives. And that goes back to the histone methylation modification. And it's very interesting to see how one drug can affect the entire process. And even though this has not been tested in humans, many people that have reviewed these results can almost say that this is probably exactly what is happening in humans when their host, for example, the pregnant mother, also is using such psychostimulants during the pregnancy. That's all I have for this video. I hope it wasn't too long. Thanks for watching.